Yeah, thanks to the organizers so much for this opportunity to speak. And I'm really glad I'm speaking directly after Katie because I'm talking about some similar things. And some other folks have touched on some concepts that I'm going to talk about as well. And I'll try to keep it relatively high level. But we in our lab, which our lab's relatively young, I've been at WashU for about 3.5 years now. But we think a lot about chromatin and epigenetics. We think a lot about transcription factors. We think a lot about how they interact. And specifically, we have hypotheses about how those interactions might impact cell fate. But just to like kind of frame the question. We operate under this principle that transcription factors are sort of determinative agents of cell fate. This principle that all of our cells in our body essentially have the same genome. Sorry. Yep. Okay, there we go. Essentially have the same genome, but they're regulated in different ways to produce different cell types. Is we're really interested in that process. And I think one of the most instructive things about, and again, I'm glad Katie introduced all this. One of the most instructive data about how transcription factors are important is the fact that you can actually express them exogenously in different cell types and convert to cell types of interest. And I really like the example of how fibroblasts, given one set of transcription factors, MyoD, can convert to myoblasts. Given another set of transcription factors, this is sort of your classic BAM cocktail, but there are other cocktails for different types of neurons that Katie introduced, can make neurons. So a single cell type, given the direction you push it, can acquire different states. And I think the exciting thing about this is we know that transcription factor and TF regulatory logic is relatively well defined. We know a lot about how transcription factors access their target motifs in the genome. They bind those motifs and then recruit cofactors and coactivators to stimulate the activity of constituent genes. And so the fact that it's well defined, as this audience knows very well, means that we can set up these gene regulatory networks. We can understand the exact input-output conditions by using various techniques like RNA-seq to measure expression level of TFs, ATAC-seq to measure how the binding sites that they bind are rendered accessible for activity. And then we have a variety of databases where we've elucidated in vitro and in vivo the specific sequence determinants of where transcription factors bind in the genome, what sequences they have affinity for. And so using all this information, we can do all kinds of fantastic predictive tasks, including predicting cis-trans perturbation effects. If you were to change the sequence, what that effect on the GRN is, if you were to remove the transcription factor, what the effect on the GRN is of that. And there's a tremendous amount of work that I can't even begin to count, that especially a lot of it in the past 5 years, that has borne on these things. But going back to this idea of direct sort of cell fate conversion, I'm really interested in knowing, can we actually predict, if we were to take a GRN and apply a particular TF, could we in silico predict where it's going to go? And I think the answer to that is probably not yet, particularly because it's well known, again, that a lot of these cell fate conversion trajectories through direct TF reprogramming are relatively inefficient. You got a lot of off-target cells. You got a lot of cell death in a lot of cases, supposing that there's something about the cellular state that is not receptive to your ability to transition to the fate that you want. So the hypothesis here, and I think you could certainly challenge this, but it's the hypothesis that I'm going to state, is that there are cell intrinsic or cell autonomous states that are instructing this to some degree, right? And I'm also going to hypothesize, since I'm relatively biased in this regard, that it might be encoded in chromatin in some way. So we think about chromatin a lot. This is kind of what it looks like in vivo or in situ. In every eukaryotic genome, they're littered with and really saturated with nucleosomes that DNA wraps around, roughly 150 base pairs and 1.5 turns. This is what it looks like in structural form. And importantly, these 8 histone subunits that compose the nucleosome are chemically modified in various ways that can, in principle, affect the sort of stringency with which DNA is wrapped around the nucleosome. So it changes the ability to sort of reveal or render those sequences accessible for activity. And this is critically important because essentially chromatin acts as an intervening barrier to a transcription factor's ability to effectuate its function via binding to its target sequences, right? I think we sort of conceptualize this generally as chromatin state. And for the uninitiated, that you could quantify chromatin state, and depending on how you characterize it, there could be hundreds of different chromatin states. But in many cases, we sort of boil it down to two basic states. One which would be more accessible or considered euchromatin, where this is sort of your active regions where you have your TFs bound. You've recruited your coactivators, you're stimulating transcription. It's associated with specific post-translational modifications that are associated with more accessibility and more mobile chromatin. And then you have sort of your repressed heterochromatin that is more tightly wound, more tightly compacted. It's perhaps less mobile. It's associated with other different chemical states of these that are modifying these histones. And so in many cell fate conditions, you're at any given locus, you're going basically from here to here, right? From repressed to active. And so the degree to which the transcription factor is able to sort of traverse this boundary is very relevant to whether you're able to actually ultimately convert cell fate. Yes.
It's partially correct, right? Because what you see is that most of the genes are on euchromatin, even when they're not active.
Mm-hmm.
It's not like when they become inactive, they switch to heterochromatin. You see, the location of the genes, active or not active, they're most on euchromatin, and the heterochromatin has very few genes. So,
I mean, I think
it's not like the switch that you have. You go from mid late to acetylate, and it's.
I mean, I think probably don't take this as literally as you're taking it, right? Like we can characterize chromatin.
I'm just looking at the picture.
Yeah, no, I understand. Yes, but point taken. I think you can characterize chromatin states in a lot of different ways. It may not be specifically modifying in a particular way like this, but as I'll tell you, we are interested in how particular modified states might influence.
There are a few of them that works like that. That's correct.
Yeah. Okay.
But even at the single gene level, if a nucleosome is sitting over the promoter, it's off. Right, like nucleosomes are repressive. So
yeah,
if you wanted to,
I understand that. I can go over that discussion forever. I'm saying that if you just look in general, all the genes that are in some particular cells, independent of which cell type you look at, some cell types have some gene active, some genes not active. Even if the genes are inactive, they tend to be on euchromatin. They need additional regulation, addition to be on euchromatin to be active. So I'm saying you're going to find very few genes. It's not you're going to find none, but there are many less genes on. It's not like if you get your particular cell, the genes that become inactive, now they move to heterochromatin.
Yeah, I think relevant to that, that's totally correct, right? That there are genes can be, if we're considering activity to be like RNA expression, right? There are genes for which we don't see RNA expression that are not embedded in heterochromatin, right? Yes. However, I'm interested in non-heterochromatin states too, right? This is just sort of an illustration of one way it can work, but no, I appreciate that clarification. So I guess the broader question here is, and I think it was Brian in the first talk who brought up this concept of constraints, right, on ability to convert cell fates, and I think I do think of chromatin is at one flavor of constraint on where you can go from a fate perspective, right? And so can we characterize how chromatin sort of sets up constraints or informs or perhaps instructs in some way the ability of TFs to bind and then effectuate their gene regulatory functions? And just to tell you about how we try to answer these questions, we use a collection of techniques known as chromatin profiling or epigenome profiling, which is very simply that in the cell, we take antibodies to the modifications or chromatin-associated proteins that we're interested in, use them using various enzymatic means to enrich for the DNA at which they're bound, and then basically sequence it. So so we can understand at a locus-specific level exactly which of these characteristics are associated with different gene regulatory states and how those are different across different cell states and cell fates and how they might inform how we think about gene regulatory architecture and how that changes across fate, right? And so one thing that I've really been grappling with is that, from a qualitative perspective, we know that chromatin kind of impacts TF function in some way, but it's very challenging to actually quantitatively assign sort of a number to how does chromatin state X influence the ability of transcription factor Y to regulate gene Z, right? Precisely because gene regulatory state and chromatin state are reciprocal, right? One influences the other sort of reciprocally. So it's very difficult to disambiguate those things. And so I sort of formulate this in the paradigm of sort of a competitive inhibitor, where if we think about sort of an enzyme that has an active site that is inhibited from sort of the binding of its substrate, we can sort of formulate that in the case of chromatin, where your nucleosome is your inhibitor of your DNA motif binding site, and the TF is your substrate in this case. And so from that perspective, we can sort of think it, and bear in mind, I would characterize myself as mostly an experimentalist, so I'm not, I wouldn't say I'm primarily a mathematician or computational biologist, or really not a mathematician at all. So I'm really happy to be here, but yes, please, I'd like any thoughts on these, the way I'm conceptualizing this would be really welcome. But if we try to model this, basically, what we think is that there are kind of three variables that we need to characterize here that we need to disambiguate. One being the TF expression level, which would sort of be the concentration of your substrate. One would be sequence context, which dictates your inherent affinity, because there's sort of an in vitro affinity that you can calculate between a transcription factor and its target sequence. And then crucially, we need to be able to separate the chromatin context from whatever's happening with binding and gene regulation. And I'm going to focus on TF binding for the majority of this, because that's a lot of what we focus on, but you could extend this to the regulation of individual genes, and you could further extend it to the collection of gene regulatory changes that impact cell fate, right? And so what we've sort of landed on as an experimental strategy to try to acquire these three different variables is to basically use different cell types as a reservoir of different chromatin states in order to challenge the same sequences in the genome with different chromatin states across different cell types. And then if we introduce exogenous transcription factors and express them at specific levels, then we can calculate, okay, at a particular, and again, these imaginary diagrams that Katie introduced, I'm really happy you were right before me, but we can actually, if we do a good job of modifying transcription factor expression level, then we can actually produce these sort of input-output plots where we say, okay, at a particular transcription factor expression level, do we get binding in this specific chromatin state? Are specific chromatin states receptive? Are they inhibitory at a sequence-specific, locus-specific level? Or does it not matter, right? Yes.
I guess, do you notice differences depending on what types of transcription factors you use? Because
yeah, that's a great question.
Because if you have a pioneer factor, that's going to work very differently than.
Yeah, we think a lot about pioneer factors, and there's, I didn't even want to get into it here because it's such a muddled field. But yes, I think one could use a strategy like this perhaps to assign a classifier if you wanted to say this is maybe a pioneer factor, maybe it's not.
It's okay for what you're doing, because the pioneer factors is just local on each nucleosome. It's not an effect of full exposure, not exposure. So
yes, in the classical definition, it's whether it can engage with a nucleosome and then displace it, right? And that's sort of independent of the chromatin states that I'm formulating here. So I totally agree with that. And so that's part of why I didn't want to introduce here, but the point is well taken. Like, different transcription factors are certainly going to respond in different ways, both because they might have different types of interactions with different chromatin states, but also because obviously the affinity of their various binding sites is inherently different, right? And so that's an important characteristic to keep in mind. Yeah.
When they're hierarchically organized, like FOXA factors in the presence of NKX21 do one thing, in the presence of HNF4A do another thing, SPDS, PINK4, there's a hierarchy of transcription factors.
Yeah.
How do you conceptualize?
Yeah. That's sort of like, I think we're trying to break it down into constituent parts here. And so trying to analyze individual factors in isolation. One thing I will say is that what I'll show in a little bit is that you can kind of get at basically cooperativity by identifying whether a certain site is already accessible or not, because that tells you whether a transcription factor is probably bound at that site. And so that could tell you whether a particular state is synergizing with another set of factors or something like that. We haven't done a lot to push on that too much, but for this particular approach, it's kind of like, let's take a single factor, figure out, you know, what its receptivity is to interacting with different chromatin states, then move on to the next and the next, and then from there perhaps start to get to more cooperative aspects of it, which the cooperativity is extremely important, as has been elucidated by a ton of work. But yeah, we're kind of leaving that a little bit aside for now. Yes.
Quick question.
Yeah.
The green curve on the top.
Yeah.
Are you implying a hysteresis? Is that green meaning you remove? It's not only cooperativity that complicates, it's also the inverse.
Yeah.
Because chromatin state affects transcription and transcription affects chromatin state. And the only way to get this hysteresis loop you see on the top.
Yeah, I don't think that's specifically what I meant to infer with that. I think it's more that the intention was supposed to schematize that there perhaps might be some chromatin state wherein if you were to take that same sequence in one chromatin state versus another, you would see a difference in transcription factor dependent activity. That's but yes. So I think one real challenge is actually expressing transcription factors at very precise levels. And again, Katie has done tremendous work on this before. Our version of this is basically we take a two-component system, one of which is doxycycline inducible, and the other of which uses this DTAG degron system. So whereas in doxycycline, we find that if you express at different levels of use different levels of dox to express at different levels, we actually get a lot of variance in the single cell expression of particular factors, both by immunofluorescence and flow. And actually, if we use dox to drive at a high level and then use DTAG to degrade at specific levels, we do a somewhat better job of expressing the transcription factors at the specific levels we want, which is critically important for modeling what we actually want to model, because we don't want a mixture of high and low expression states. We want sort of like intermediate expression states, right? And so this is work of a really talented graduate student, AJ Federico. I'm glad someone brought up FOXA1 because that is the factor that we're focusing on right now. We can actually express FOXA1 exogenously in a variety of different cell types. This is just looking at fibroblasts, human foreskin fibroblasts right now. We can drive with dox, but we actually degrade all of it if we add DTAG. If we remove DTAG, then we express dox at a pretty high level. And you can also see from the fate conversion perspective, we do start to express hepatic transcripts such as, this is protein actually, hepatic proteins such as albumin. It's at a relatively low percentage, but that's actually kind of nice because then we can say differentiate what the sort of transcriptional fate conversion efficiencies are again, and start to look at the fate perspective there, which I'm not going to focus on here, but just to say that in this system we could get at these questions. And just to say, this is sort of a data representation of what I just told you, that the variance across single cells gets quite high if you use dox only, and if you use dox plus DTAG, it remains relatively low. And this is borrowing the Gini coefficient, which is a concept from economics that tells you about the inequality across agents in a system. So AJ, okay, so I'll try to get through this pretty quickly. Yeah. So AJ used this in K562 cells just as a pilot experiment. It's not really important right now what the sort of chromatin states we're looking at, but suffice to say we can look at states that represent a variety of gene regulatory states, and then look at from a binding perspective, bound versus unbound, and then use chromatin states in sort of basically a basic random forest model to derive conditional ectopic binding probability. And we can see chromatin actually does a very good job of predicting the actual binding of the exogenous FOXA1. And we can see that there's some unexpected things we see, including that there are specific sort of promoter and enhancer associated chromatin states that, and I'm not showing the data here, but these are actually these individual states, K4 mono and K27 acetyl. We can show that they're actually independent of accessibility. So although they are highly associated with accessibility, accessibility is not specifically required to get the stimulatory effect of these modifications. So that was sort of a surprise for us. And so you can see there's monotonic relationships between the amount of a particular modality and the probability of binding. Yeah.
So are these? Do you see a downstream effect of ectopic FOXA1 binding though? Like so if you do a multi-tag experiment, do you see more K4 mono that's going to the ectopic binding sites, or this is the pre-existing?
This is the pre-existing, yes. So we have the data on the downstream. We actually try to keep it relatively short so that we're not having secondary effects intervene there. But in all likelihood, you would start to see a little bit more at some point. Yes.
What you just said, I'm a little confused. Your plot on the right. But looks like here the
yeah. So this is looking.
The attack seek appear to be correct. Connected. You said they are not connected. Okay.
Yeah, sorry. I should say that individually attack is important, right? Like if you have more accessibility, you get more binding. However, across the concentrations of different K4 monomethyl states, if you quantify K4 monomethylation states and you basically vary the amount of accessibility across those states, if we can derive from this like deep learning SHAP model that basically the importance of K4 monomethyl stays high even when the accessibility states are low at the same level.
High enough.
Yes. Yeah. Sure. Yes. I mean,
that's good. That's
yeah.
good. I don't understand.
Yeah. Exactly. And so, sorry, just to breeze through a lot of this, we can basically produce these binding curves across thousands of different loci across the genome.
That's an interesting result because the chromosome is very dynamic, so it's very interesting if these sites are opening and closing or something like, right?
Sure. I mean, and to that point, I think in some ways, some of these actual chromatin states are probably a little bit more stable than the accessibility because the accessibility can be quite variable across time.
It's an interesting result just to go and investigate some of this.
Yeah. And yeah, sorry, I'm going fast because I want to meet time, but because I would say like Lior, like I don't have to eat lunch, but actually I do. So you've got me pegged there. But so the hope is that we can kind of derive these like constants of inhibition where we see across different chromatin states. Is there a positive inhibitory effect? Is there a negative inhibitory effect? And sorry, I should say that modeling work was the work of actually a high school student at Lab Kento, who's been awesome. And just to say, we're going to do this at massive scale in collaboration with Yoav Gilad's lab at University of Chicago, using their heterogeneously differentiated culture system to try to produce 30 different cell types at once in a single culture, and then use single cell methods to be able to disambiguate this and directly pair sort of quantitative binding measurements with fate conversion efficiency measurements across different cell types. And that work is ongoing. It's being led by Jingma and Nisar Ahmed. So yeah, these are the conclusions. I won't go through them just because we're at time, but do want to acknowledge my lab collaborators at WashU and definitely Yoav and Katie at UChicago. And yeah, thanks for your time and listening. Appreciate it.